The cellular membrane is a selectively permeable barrier that there's analogous structures in all forms of life, but what's really interesting is to note that their composition varies just a little bit. For example, in the world of bacteria and archaea, there's often a greater protein to lipid ratio than in eukaryotic life. So despite the analogous function of a membrane in all cellular life, it will vary in composition. In all cases, it serves the role of dictating the relationship of the cell with its environment. And certainly that is a majority of what we are talking about when we say selectively permeable, because it is dictating what gets in and what gets out. So very much playing that exclusive role in trafficking across the surface of the cell. Now, I want to point out certain components of the cell before we look at a model that talks about the movement of the membrane. Recall that a cell membrane is based primarily on the structure of a phospholipid bilayer. Now, a membrane is different than a phospholipid bilayer in that a membrane has proteins that span the membrane, or that span the phospholipid bilayer. So notice here you can see that we have two different types of proteins. There's one type that completely spans the bilayer. These are termed integral membrane proteins. And then there are the peripheral membrane proteins that associate with the membrane on one side or another, but they do not entirely span it. So another name for integral membrane proteins actually is transmembrane proteins, because they are clear across the entire phospholipid bilayer. Notice that in all of my drawings, the top is always the exterior of the cell, the bottom is always the interior. So I won't always label that, but I want you to bear in mind that that's the directionality that is often used. So integral membrane proteins pictured there in pink and in blue, then that red one is a peripheral membrane protein. Sometimes you also have lipid tethered proteins that are somehow associated via a bond, but they are not even as intimate as a peripheral membrane protein is. So notice that we have the phospholipid bilayer, in which each one of the phospholipids has a polar head. So this region here is the polar head of the phospholipid, and then it has two long fatty acyl, very water-hating tails. So I'm highlighting those here in green at this time. So you can see there is the water-hating tail of the phospholipid, and here's the second water-hating tail. The head loves water, and the tails, they hate water. And so it makes sense that they put their water-hating tails together and their water-loving heads facing the exterior and the interior, which are very water-filled environments, very aqueous environments. Cool. So another thing that you're going to see within a membrane is a structure called a hopanoid. And these hopanoids actually are functionally equivalent to the sterols in animals and plants, that is cholesterol in animals. The purpose of these molecules is to modulate the fluidity of the membrane. That's a very sexy-minded way to say they keep it from freezing when it gets too cold, they keep it from melting when it gets too hot. So the hopanoids play that role in bacterial cells. They stabilize the membrane and modulate its fluidity, keep it from melting, keep it from freezing. We can also draw our attention to Glycol Lipids, which this is a cool mishmash of two different molecules. There is a carbohydrate, which is a polymer of sugars, and it's attached to the phospholipids. So we think of this as the term glyco refers to the carbohydrate. Like you guys know about glycogen, right? Glycogen is a carbohydrate storage polymer that we have in our liver. So glyco means the carbohydrate, whereas the lipid is the phospholipid. So together they are a glycolipid. We can also get cool conjugates of carbohydrate and protein. So we can get a glycoprotein. So imagine a carbohydrate attached to this protein would be a glycoprotein. So one interesting bit of trivia is that hopanoids are a highly prevalent compound in the Earth's crust, and in fact they're thought to have contributed highly to the petroleum stores. So that's kind of a little interesting bit of trivia, particularly for those of you engineers out there. Now, as it turns out, the movement of proteins within the phospholipid bilayer of a cytoplasmic membrane is very akin to swing dancing. And some of my brilliant prior students created this, along with the entire swing club, in fact, should be credited, this incredible video that shows the way in which swing dancing is very much the same as the movement of phospholipids and proteins within the cytoplasmic membrane. So this demonstrates the two different motions that are allowed with both proteins and phospholipids. They can rotate or they can move horizontally, like the couple who is coming across the front of your screen. However, one motion that is not typically allowed is flip-flop. Oh, like that. That typically is got to be enzyme catalyzed, so we don't usually see phospholipids flipping that way. But this model that dictates protein or a movement and phospholipid movement within a membrane is called the fluid mosaic model, and it's a really fitting name because mosaic refers to the way in which proteins are interspersed, like a mosaic. You've seen a mosaic of tiles, right? The way in which that is interspersed. Then the fluid word in that model, it refers to the lateral, the lateral motion and the rotational motion that is allowed. That is to say that a membrane is not just rigid and just no motion in the membrane, but instead, it's really loosey-goosey. There's that rotation. So there is a lot of fluidity to a membrane. Now, this model was developed in 1972 by a man named S. Jonathan Singer and Garth L. Nicholson, and it describes the membrane as a dynamic structure in which both proteins and lipids can diffuse laterally and rotate. Now, I have a cheesy cartoon to show you guys this. It's by no means is this cartoon showing everything, because I had a hard time showing rotation. My animation skills are B-rate at best, but I want to show you kind of this idea that it is dynamic, right? There's the lateral motion, horizontal. If we could show those also spinning, that would be a good thing to also show. I want you to think about the knowledge that the amino acids that make up this transmembrane protein, they're different in nature depending upon where they're located. So I want you to think about amino acids that are located here in the center of this protein, sticking out from the sides of this protein into the phospholipid tails. And I want to know, are those amino acids likely to be water-loving or water-hating? So then I also want you to think about, would it be easier to extract a peripheral membrane protein or an integral membrane protein? So in this picture, we're only showing integral membrane proteins, but what if we drew on here a peripheral membrane protein, a little green guy, that maybe is just associated with the outer leaflet of this membrane? So, hey, I just used some sexy terminology. Let's make sure we're all with me. This is the outer leaflet of the membrane, and this is the inner leaflet. So notice here this little green peripheral membrane protein is associated with the outer leaflet. So what I want to know is which would be easier to extract in the lab. If you wanted to study this little green protein or if you wanted to study this big pink protein, which one do you think you'd have an easier time getting off the cell or out of the cell? So you can give that some thought, and that'll be your second question. And then what are some functions of an integral membrane protein? So I want you to mull that over and think about what are some functions of, say, this pink protein or this blue protein. And I'm going to cheat a little because here in a moment I'm going to give you one possible function. But I want to further think about the role that this membrane plays in serving as a selective barrier for the cell that is dictating what will gain passage in and what will gain passage out. And in that way, I like to make an analogy actually to, if you've ever been to a super trendy dance club, you'll remember that the bouncer at the front door only lets in certain people. And one of the keys to getting in, particularly if you're a lady, is to be very skinny and, you know, definitely wanting to drink alcohol, right? So I think this is very much a good analogy for what the membrane does, because the phospholipid bilayer itself, okay, it's not going to let in anything that is hydrophilic, that is water loving. So it's like this membrane, and you guys are probably too young to have seen Coyote Ugly, but you know how they're chanting, hell no, H2O. Okay, well, this is what's going on here. This membrane is like no H2O loving molecules allowed in, right? Like the bouncer at the dance club. Nobody who wants to drink water, just people who want to drink alcohol. Okay, so then also this membrane is going to be exclusive based upon size. So only little, that is low molecular weight molecules will be allowed to pass. So nothing large will be allowed in. So this is where the analogy comes to full fruition, because all the ladies of low molecular weight who don't want to drink water, then they're the ones that are allowed in. So it's a lot like a membrane. So only those small hydrophobic molecules like oxygen and carbon dioxide, those are the only things that may pass by simple diffusion. Other molecules have to get in using a special transporter. So there has to be some protein that is dedicated to allowing in these other molecules. So one of the functions of certain integral membrane proteins is actually in energy generating. So in energy generation systems, we see these integral membrane proteins playing a very important role in transferring electrons and pumping protons. So in prokaryotes, some of them are capable of something that we call respiration. Some of them are capable of photosynthesis. And in those particular microorganisms, spanning their membrane, we're going to see these integral membrane proteins that are involved in energy generation. And what they do is they transfer electrons throughout the members of this chain. And as they do so, they use the resulting energy to pump protons. So those prokaryotes capable of respiration or photosynthesis have the compounds of the electron transport chain across their membrane. The compounds of the electron transport chain shuttle electrons and eject protons. So this is actually a picture of the E. coli electron transport chain, where you can see that it's shuttling a bunch of electrons and it's going to pump protons across the membrane. So notice here that a proton gradient begins to be established. This proton gradient is called a PMS. I think you guys are just about to meet my cat, Cal. So this is Cal. Cal is currently telling me he very much wants to be on the vodcast. So perhaps his energy generating systems are particularly on high throttle because he seems to have a lot of energy today. So why don't we dissect this a little bit? So let's say, and I'm going to just get Norbert. And I don't want to niche Norbert. I mean, I don't want to make Norbert necessarily into E. coli. But let's say that Norbert is E. coli and that across the membrane, which is actually shown in yellow right here, across Norbert's membrane, we'll say that there are integral membrane proteins spanning that. And as the electrons pass through those integral membrane proteins, the energy released from that pumps protons. So out here, outside of the cell, we begin to see the establishment of a gradient. This is like a pent up battery-like power. This is the PMF. And by analogy, this is a lot like the energy that we get from a dam, right? Where we pent up a bunch of water behind this dam, and then we can tap into it to generate energy, to turn turbines, to make those turbines run, to make electricity, right? It's a conversion of energy. That's what we have here going on in Norbert. So imagine this pent up battery-like energy all around it. It's almost a buzz with energy, right? And now when this bacterium needs the energy, it will allow some of those protons to rush in. And with that inrush, those protons will power something like the way that the water behind a dam powers a turbine. For example, for Norbert, one of the things that pent-up battery-like energy can power is the rotation of a flagellum. So as the protons rush back in, the energy from that can be tapped into to allow for the rotation of Norbert's flagellum. And that rotation, of course, can allow the bacterial cell to either move in a forward direction, or to tumble, to kind of change direction. So this is one thing that PMF powers. Another thing that it can power is the formation of ATP. So across the membrane of Norbert, there is a structure called ATP synthase, which taps into that PMF and converts the mechanical energy into chemical energy, forming the ever famous ATP molecule. Lastly, this energy of the pent-up PMF can also be used for transport. So say that there's some molecule like lactose outside of the cell and Norbert wants to get it in, then it can utilize the PMF to be like the river that flows downwards and into Norbert and carries the lactose with it. Now, you all have seen Finding Nemo, right? There's that section with the turtles, and they're getting to Australia. They're riding the EAC, dude, right? And that's what I think of when I think about the use of the PMF to drive inward transport. It's like ride in the PMF. So if it's that lactose molecule that wants to get into Norbert, then it can flow downwards, downstream along the PMF current, and that's a way to transport molecules then. So the electrochemical gradient provided by the proton motive force can be used to synthesize ATP and also to drive certain other cellular processes you might just want to jot down there, such as flagellar rotation and transport. Let's end our coverage today with a quick practice question that will allow us to review our knowledge of the fluid mosaic model. So looking at this picture, we can see that we've got a membrane with peripheral membrane proteins, we've got integral membrane proteins, and a somewhat labeled phospholipid bilayer. At least the region with the tails of the phospholipids is labeled C. So let's see if we can take this picture and use it to help us answer the question. The protein labeled B is a peripheral membrane protein. Yes, that looks to be the case because it is associated only with the inner leaflet of the membrane on one side of the membrane, in the intracellular side, so it is a peripheral membrane protein. Unlike A and D that are integral membrane proteins, right, they span the membrane entirely. So according to the fluid mosaic model, the protein labeled A is free to diffuse laterally and rotate. Yeah, we know that it can definitely swing dance, can it? It can move in a horizontal direction. It can also rotate. It doesn't do a whole lot of flip-flopping, right? That's not so much of an allowed motion unless there's a crazy enzyme out there to catalyze it. So according to the fluid mosaic model, this membrane is rigid, inflexible, with sterols as the primary component. Wow, there is truly nothing that is correct about that statement. Pretty easy to select that as the false statement, right? We're looking for the false statement, so pretty easy to say that C is, because remember that a membrane, by definition, the fluid mosaic model, says that it is dynamic, where there is allowed rotation and lateral diffusion of both phospholipids and proteins. So we would say that C is clearly out of the park, false. Let's just make sure we understand why D and E are true. The protein labeled D is a glycoprotein? Yeah, it is, because notice here's the carb region. So this whole polymer right there, that is a chain, right, is a carb. And then this is protein. Cool. So then, although not pictured here, many bacterial membranes contain haploids, which play a role in modulating fluidity. You bet. So only one clear false statement there. I think we can end the day feeling soups good about ourselves, and I'll see you all very soon.